I would like to talk about is to prove again and summarize what was said during the program, the session. We can't change our genes, but we can manage them. And the most important factors of such management is physical exercise and our food. And if we know now microbiome, because microbiome can't be ignored anymore, it's a very important factor which influences many parameters, practically all processes of metabolism. And the last point, several ideas that we would like to share with you, how this knowledge can be used in clinical practice. Today we've heard several very interesting papers. Omics Cascade is in the middle of this field of medicine, and we can create possibility for prognosis of how certain disease is going to develop in a certain person. Of course, we can't use it in clinical practice yet to the full extent, but we always direct our clinical thought in this direction. Today, there were several questions about importance of different SNPs, but I wanted to say that there are two different genomics, structural and functional. Of course, structure is inherited, but all this cascade which is, of course, it's expensive, complicated, but it exists exactly for this purpose. How exactly, how precisely we can change the functional part of our structure. And if it's possible, I think that you saw today in the presentations that were presented here, there are questions on what healthy diet is better than another healthy diet. Then we won't have these questions, because this genetic uniqueness, biological uniqueness, and knowledge of profiling of our patient will give us the answer. The possibility to research microbiota gives us another tool for personalization of our approach to our patients. Phenotype is very flexible, and what we can suggest to our patients, the earlier the better, and the better results we'll expect. We talked today about main epidemic signals that can be changed, and its long-term matter. It's the problem of choice of our patient, and it's one of the. And also we can measure quite precisely certain parameters, which should be then the factor of choice for our patient who wants to follow the directions. Here we show you two twins as a proof of how plastic the phenotype is. And also there can be that someone is very healthy but dies in the gym, like James full of X, or Mick Jagger, who has no claim for healthy life, but who is still alive and healthy, considered comparatively healthy. For the moment, we know about genes which are in charge of our metabolism. They are well researched. It's not difficult to do this research in the laboratory, but to use this information that was got in genome laboratory, it's very important to remember to know about transcription regulatory factors that are realized, taking into account such important factor as nutrition. Nutrition has immense importance. Our cells can realize and carry out the processes of energetic metabolism, but what path, what way they're going to choose is dependent on transcription factors. And the nutrition recommendations on diet, on nutrition, on exercise, they work. But they work only in the case if it is used long term. It's not some acute change that we can do for short time. Microbiome is a very important factor, as we all had a chance to understand today. And how the change in microbiome can influence certain diseases. It's a very interesting question, very interesting topic. I'm sure my colleagues will agree with me. And of course, it's only the very beginning of the research. And the phrase that was already, everyone said this, that bacteria is something that we ate before, and their nutrition is also connected to our life. Some information about relation between genetic structure and manifestations of these genes that are involved in metabolism and food signaling, and also metabolic function of bacteria, just to prove once more that it's really working substrate of really working thing, that we really can, probably we can rely upon it. I would like to show you that with different diets, detoxification is expressed in very different ways. For example, if there is a diet with high fat, low fat, balanced diet, we can see how the intensity of processes is changed, and it's the whole spectrum of different genes that are involved in these processes, connected to immunity. And it's also proved that different types, including fat diet, have completely different consequences on immune function of the gut. And there is very interesting marker. I don't have enough time to talk about it in detail, but acrylic receptor that at the moment is recognized is fundamental factor of transcription, which influences quite wide range of metabolic pathways and toxification, inflammation processes in the gut.
Противовоспалительный эффект пищи имеет абсолютно подтвержденные молекулярные механизмы. В основном это лиганды, которые связывают.
These are ligands that bind to an acrylic receptor, which finally leads to expression of genes that lead to immunity modulation in the gut. These are mostly ligands that are of nutrition provenance, like middle 3 carbinol and then cruciferous, yeah. So the cruciferous plants like broccoli, cauliflower, so all that family of plants, flavonoids and polyphenols. So these are anti-inflammatory nutrients, which being ligands of acryl receptor lead to stimulation of a whole number of immunity factors, including interleukin 22 with its great metabolic role. Then there are substances that are also ligands to this of this receptor, tryptophan, another anti-inflammatory molecule, which in the end breaks down into a ligand on that same receptor. Tryptophan is directly involved in the synthesis of anti-inflammation substances, and plant fibers, which again break down in the gut, activate serpentines, that lead indirectly and through the action of acetylase to an anti-inflammation effect. Just a couple words about fibers, because we understand that this is very important for healthy food, and it's interesting to know that their epigenetic activity is different in each case.
Производству разного синтеза КЖК, и как следствие имеет абсолютно...
They have different metabolic activity. Some features, some characteristics of the fibers. How can I go back? I'm using an abbreviation, guaric tar, arachidonic and resistant starch, as different types of fibers and a control group. It was very interesting to look at the regulation of expression of genes by these 4 fibers, and separately there is a resistant starch whose influence on expression is like a control group. There are certain specific characteristics that are typical for each of the groups of fibers. In my opinion, it's another very important characteristic that makes it possible to establish certain correlations, including correlation in clinical practice, is the connection between gene expression and liminal microbiome. I think this picture makes it evident that there is correlation between clusters of various genes of different functional characteristics and the clusters of bacteria of different functional characteristics as well, which means that if we can confirm that in humans, then perhaps it could help us evaluate how expression changes, in which groups of genes we see a dependence on certain strains of bacteria which we can group into clusters. Of course, the program is the main regulator. Another interesting fact, the microbiome of the gut, which we often see as normal, quite often it does not produce its positive functions. We all know that red meat leads to rectal cancer, but mice who receive a different diet, they have compensatory hyperproliferation, which can also become malignant sooner or later. Mice who have the same diet and antibiotics, their epithelium is not damaged. Researchers explain it with the presence of this mucolysis, with the presence of reducing bacteria, mucus-reducing bacteria, who prevent the degradation of mucus, because these bacteria induce the development of cytotoxic mycelia, whereas with antibiotics there is no such effect. I think this is not the right audience to speak how important microbiome is. Usually we call it microbiome now, with the possibility to sequence the genes. In clinical practice, even now, when we study the genome of the microbial genome, we can make certain profiles, we can establish activity, we can measure vitamin genesis and a certain enterotype. Enterotype is like a blood type. We are all different. There are 3 enterotypes at present, and as a result, each of us belongs to a certain signature with its risks and its metabolic profile. I've been listing this fact to share with you our idea of trying to collect all the data there are and to try to implement in our clinical practice to understand how human metabolism is actually constructed, how personalized nutrition can be used, where is the border between health and sickness, something that Ancha has mentioned. I will give you an idea of a research that we would like to invite you into, because of course we'll be telling a detailed protocol to those who will be interested. The goal is to create a systematic approach and search of metabolic reasons using genomic technologies, but the main goal is to describe the axis of microbiome and gut function in normal conditions and pathology in humans. The more patients, the faster it will be possible. Another task is to establish the markers of metabolism and functional status and phenotypically, genotypically stratified subgroups of patients, divided by their genotypic and functional status. And third, find risk-inducing alleles and diseases. And finally, evaluate the predictive potential of existing genetic markers that are widely used in clinical practice, both their positive and negative predictive value. As you see, this research aims at answering the questions that have been raised yesterday and today, now and again. Something very important here, of course, a few SNPs are not enough to make a diagnosis. This is just a predictive story, as Ancha said, and I just wanted to confirm it. This is something that defines the reaction of a patient. Is it necessary? This is just the normal reaction and nothing more. But it's necessary if we want to conduct some long-term research. Going back to 360 degrees research, which we invite you to participate in, this research will be based on gene typing and sequencing the microbiome of all patients. Certainly, every patient will be questioned and functionally studied and so on. There will be 2 stages. Stage 1, we want to research the function of the gut and the connection of the functional status of the functional analysis of the patient with his or her microbiome. The idea is studying the function of microbiome, which we all know, using the markers that are the golden standard and which explain certain conditions which we can find. For each of these states, there will be correlation with gut microflora, and as a result, we hope to see to what degree the knowledge about microbiome of the gut correlates with certain functions that are well known to every clinician. Which functions are these? Well, constitutional resistance, of course, digestion, immune system work, motor functions, enterohepatic function of bilous acids, vitamin synthesis and exchange, absorptions, mediators, barrier functions, inflammation, detoxification, supplying substrates for lipoglucogenesis, lipid exchange, food intolerances, and contaminations with fungi, with worms, and so on. Another important thing, we will not be taking patients who are in acute state or an acute phase of a chronic disease. So basically, we're working with functionally or conditionally healthy at the moment of their inclusion. And as a result of stage 1, we plan to receive detailed reports about microbiome and metabolism based on the markers that we talked of. If at this stage we will find certain pathologic changes that need pharmaceutical correction, that means that those patients will leave the cohort. But for the majority of the patients, knowing their microbiome will suggest certain food dietary recommendations, metabolic correction, if necessary. And the next stage of the same research, stage 2, will be solving the task of personalization in accordance with a specific genotype, because at this stage we were planning to stratify the patients depending on their gene markers of functional status and the microbiome, and then monitor their condition depending on the group they're fitted in. We will study not the diagnosis, because these are patients who are not sick. We will study to what extent their functional state based on genetics, questionnaires, and so on leads to certain metabolic, the disruption of certain metabolic pathways that can occur and become a disease, but can also go the other way, because we plan to conduct biological correction, especially as regards diet. This is the list of all metabolic pathways which we're planning to evaluate, and of course there will be separate methods of evaluation for each of the pathways. As a result, we expect to stratify risk groups based on genetic markers and the condition of the microbiome of the gut, and a personalized diet with preventive metabolic correction. Nutrigenomic is very ambitious. We basically want to prevent a disease by choosing a personalized diet. And this is my last slide. The Biomedical Center Atlas invites you to cooperate in this research, in this study. And I'm sure that anyone who wants that, they can address me